Alright, so what I'm going to do, the purpose of this is to go over what the objective of the lab is for tomorrow and go over the calculations that you need to do pre-lab and how you need to prep your notebook. So first and foremost, we're going to be using a spectrophotometer tomorrow in class. If you don't know what a spectrophotometer is, it has an energy source or a bulb that basically produces light, and the light passes through a system under study. What we're going to use, we're going to use a cuvette, which is like a little tiny plastic test tube, the one we're going to use. So light's going to pass through, but depending on the number of particles in the system or in the cuvette, let's say this is the cuvette right here, number of particles in the system, what happens is as the particles hit substances that are in the test tube, the light doesn't pass through. So what's transmittance? Transmittance is when the light passes all the way through. That's T. Absorbance is when the light molecules don't make it through the cuvette, or because they're running into molecules of dye, because we're doing Gatorade dye tomorrow. So we measure transmittance is the amount of light that passes through by putting it into a spectrophotometer. That's the whole point. But we can find absorbance because they're inversely related to each other, because as transmittance decreases, absorbance increases. And the relationship is the greater concentration you have of molecules in a test tube, as you send the beam of light from the stimulus through, the less transmittance you're going to have, the greater the absorbance. So absorbance and concentration are directly related. This goes back to, like, if you look right here, here's an example of the relationship. It's absorbance is equal to the negative log of T. So our light source right here is firing out some stimulus. B in Beer's law is represented as path length. It's the difference that the light has to travel, or the distance the light has to travel through the cuvette or the test tube that we're using. And then the concentration is related to the amount of particles that are in a cuvette. So that's related to Beer's law. Beer's law is a mathematical relationship between absorbance. So A is absorbance.
Miss Matthews, Charlie Matthews, you're needed at the bus loop, please. Charlie Matthews.
So A represents absorbance. The absorbance is related to the absorptivity, which is basically depends on the type of dye you're using. Like, for example, blue dye has a certain specific, depending on its concentration, a specific amount of light it absorbs. All molecules are different. So absorptivity is related to what type of molecule. B is represented by the sample cell or the cuvette, the path length, the distance the light has to travel. For us, it's going to be a centimeter. And then C is concentration. Since B is constant in everything we're using and E is constant, there's a mathematical relationship between A and C. The amount of concentration has a direct impact on absorbance. The greater the concentration, the greater the absorbance. What we can use Beer's law for is, if we know the absorbance, we read that, we know what the absorptivity is, and the path length, we could solve mathematically plugging in Beer's equation. They ask you to do it on pre-lab for question number 3. They'll ask you to solve for what the you know what the concentration, you can solve for either the concentration or the absorptivity, whichever one they give you. So one of the things they talk about in the pre-lab is using lambda max. If you're going to use a spectrophotometer, which is basically a machine that reads the amount of light passing through something, one of the things we need to use or find is called lambda max. Lambda max is the wavelength at which maximum light is absorbed in a sample. It's different for different colors. For blue food coloring dye, the lambda max is 630, because we look at 600, there's 610, 620, 630 represents the lambda max. That means the best place to test it for its absorption values is at 630 nanometers. So it should be nanometers on there. It's not, this is nanometers, but it's basically 630 nanometers is the wavelength of light that you have the greatest absorptivity, or the greatest amount of light is absorbed. So that's basically when you go to set your spectrophotometer, when we go to do a reading in the lab, you want it to set it to 630. Now, what do you have to do for pre-lab? One of the things is you're going to prepare a bunch of solutions. You're going to take a stock solution, which I'm going to give you. You're going to take a certain amount, put it in there. So you're going to basically put a sample of stock. So let me change the color. Red would represent the stock solution, and then you're going to fill the rest with distilled water. You're going to make a series of dilutions. Remember, this is the dilution equation we learned in Chapter 3. This represents the stock solution for molarity and volume. This represents the dilution. This has the new volume on there. New volume is listed there. This is the original molarity and the original volume of stock. So what you have to do is you're given the original value of the stock solution. The molarity is 7.0 micromolar, and it is 10 milliliters of stock solution. So you have those two values right here, right? Then what you're going to do is you're going to combine different ratios. Like the first one, you could combine 2 milliliters of water, 8 milliliters of stock. So the value of the stock would start out as 8 milliliters. You would put here. That's going to be your V1 right here. Your V2 is going to be the total. It's always going to add up to 10. So when you're doing your lab calculations, the new volume is always going to be 10 milliliters. In each of these, if you add them up, if you'll notice, they'll all equal to 10 milliliters. So the new volume is always going to be 10 milliliters. What you're trying to solve in all these is you need to solve the new molarity, the M2. You need to solve it for B through G. The last one is water, so you don't really have to do it. It's our blank, which we use to zero out the spectrophotometer before we start. So when you're doing your pre-lab calculations, just remember this. This is going to be your original molarity. Your volume of the stock solution is going to get dropped down here. The new volume every time is going to be 10 milliliters because you're making 10 milliliters of a dilution. You're solving for M2. That value is going to go up here in all of these places. So that's number 2 on the pre-lab. Number 1 asked you what a lambda max is. Lambda max is the wavelength of light where you have maximum absorbance. You'll fill this out in your pre-lab. Remember, the pre-lab always is done on a loose leaf piece of paper. You would recreate this exact grid or data table with a ruler, definitely with a ruler, or print it out and cut paste it, whatever you want to do, onto a piece of loose leaf paper. What's the point? Why are we doing this? This is called a standard curve. We are going to construct a standard curve, and we do that by taking a spectrophotometer. We know the concentration of the dyes because you calculated them, each of them in here. So you're going to fill this in by doing calculations right here. So you know the concentrations of each of the samples. Those are known concentrations. We will be able to put them into the machine and read the absorbance. So the idea is you're going to put each of the samples that you run into the machine, and you'll read the absorbance off the machine. The absorbance is directly related to concentration. So the more concentrated it is, this is not blue dye like we're doing. This is the yellow sample. That's why it looks a little different and the values aren't the same. For example, the most concentrated one is going to have the highest absorbance. That makes sense. So what you'll do is you're going to get an X and a Y in your data table. You'll have concentration, which you calculate out in your lab, part of your pre-lab. Absorbance, you're going to read from the machine. If you have X and Y, when you do a scatter plot, you will get Beer's law is a linear equation or a linear function. It produces a line. So what you need to have in your data table is your stock solution is going to be tube A. You're going to make 6 tubes, B through G. These are the ratios that you're going to use. You're going to mix them tomorrow using a transfer pipette to pipette water and dye. You're going to have to calculate out, you'll have done it for your pre-lab already, you'll fill this in, and then we use the spectrophotometer to gather this data. Once we gather all the data, you'll fill this in. You can read it off the machine, %T, transmittance, and absorbance. You can calculate it. You fill out this data. This data table has to be created in your lab notebook. So first and foremost in your lab notebook is always the purpose. The purpose of the lab that we're going to do, you explain what that is. Then you'll do a modified procedure. That's where you take the procedure and change it into a numbered list. You can shorten it, make it a little bit more concise. The next section are the data tables for your lab notebook. This would be the first data table that you're responsible for. You will have completed this part of it for the pre-lab. You can actually fill the data in. This part will still be blank. We'll fill it in tomorrow, and then we'll use Microsoft Excel using this as our X and this is our Y to create a standard curve. After we create a standard curve, I'm going to give you Gatorade. The purpose of this lab is for you to tell me how much dye is in Gatorade. So I give you Gatorade, and let's say you tell me I give you a yellow Gatorade. You tell me the absorbance is 0.6. You read it. The purpose is you take the absorbance, trace it till you hit the curve, and drop down, and it will tell you the exact amount of dye in Gatorade. That's what we do in a chemistry lab. The purpose is to determine unknown concentrations. So the overall purpose in the lab tomorrow is to determine the unknown concentration of Gatorade by developing a standard curve using known concentrations, reading them in a spectrophotometer, making a linear plot, and then testing the unknown. And that's what we're going to do tomorrow. Your job, number 1 is answer pre-lab questions 1 to 3 on a loose leaf piece of paper. So you do the pre-lab questions. Make sure you get those done. Second thing you want to do is you want to prep your notebook with a purpose. I've talked about it. You can read it in there. You can read it in the overview. So you have a purpose. Make sure you title it on the page. Third thing you need to do is a modified procedure. So a modified procedure is basically a bullet list of the amounts you have to measure. Anytime you're massing something, whatever, you highlight that stuff and list it. Make sure it's in numerical order, vertically listed. The next thing you need to do is prep your data tables. You only have one data table you have to prep. I showed it to you. It looks, I can spell table. It looks like this right here, this. So if you look right here, this is what you're going to hit pause. You can create it in Excel if you want, whatever you want to do. You can create it. If you're going to hand write it, make sure you use a ruler. The lines need to be straight and it needs to be evenly spaced out. Every data table should have its title. Your title will be FDC Blue number 1 because that's what we're testing in our lab tomorrow. All right? That's an overview for the lab. I will see you in class. Have a good day. And make sure you come prepared to class with your lab notebook.